Good morning, everyone. Thank you for joining the third DRYvER webinar. So this one is about predicting changes in ecosystem functions in DRNs. It will be presented by Nayara Lopez Rojo and Ruben del Campo. So I will first let the floor to Nayara. So I will share my screen. Can you see my screen?
Yes. Can you hear me?
Yes.
People can also hear me? I suppose that yes.
I think so, yeah.
Okay. So thank you everyone for being here. I am Nayara Lopez Rojo. I am currently working at the French Institute of Food, Agriculture and Environment in Lyon. And me, together with Ruben del Campo, will be presenting part of the work we have been doing in the work package 3 of the DRYvER project, that it's linked to the changes in ecosystem functioning. Next, please. So at first, I will be doing a general introduction about the ecosystem functioning in DRYvER networks. Then I will present the general study area and the part regarding the greenhouse gases emissions. And finally, Ruben will be presenting the results regarding the leaf litter decomposition and do like a final summary conclusion section about the general presentation. Next, please. Next. So as you may all know, rivers contribute disproportionately to the global carbon cycle. In fact, the dendritic structure made them perfect for draining all the carbon from the catchment, and so the rivers receive huge inputs of terrestrial carbon that could be either stored in the sediment. Another part will be also transport downstream, but here in this presentation will be focused in the part that is processed by invertebrates and microbes, decomposing the leaf litter and then invested in secondary production, and the part that is respired for all the organisms and released as CO2 and CH4, which are the main greenhouse gases. And in fact, we have to take in account that this is a huge part of the total carbon. And in fact, it means a total of 1.8 petagrams of carbon per year. Next, please. And especially in the context of this project, we have to keep in mind that in between 50 and 60% of the total global stream river length is intermittent, non-perennial. That means that dries at least one day during the year. Probably some of you have already seen this map, and we know that this is not a local phenomena, but it's like globally distributed. And this drying is becoming more frequent due to climate change, and it's altering both the ecosystem functioning and the biotic communities. Next, please. So in fact, drying river networks, or DRNs, as we will refer by now, are very dynamic ecosystems both in space and time. And in fact, they can have different kind of habitats, like flowing rivers. Then these rivers can dry, but can also remain with some pools. And this mosaic of habitats creates like a patchy river network that is very dynamic both in space and time. Next slide, please. Here you can see a picture to illustrate this very graphically. These are two photo traps pictures in the Albarine River network of an intermittent section. You can see that there is only 1 day of difference between them. One is for the 3rd of October and the second one is from the 4th. And you can see in the first one that the riverbed, it's like totally dry with some leaf litter remaining in the riverbed. And then the river suffered a huge reverting event, and in the second one it's totally flowing, and this like transport downstream all the leaf litter and disturbs the natural pattern of the river continuum concept. Next, please. So in fact, this phenomena of rewetting and drying events make DRNs be like biogeochemical reactors in which drying, as I have explained, alters the transport of the leaf litter, the storage, and with flow resumption, this can boost either the decomposition process and also transport downstream a lot of organic matter. Next, please. And we also know nowadays that the riverbeds also emit greenhouse gases when they are, so the rivers also emit greenhouse gases when they are dry. But there are some papers acknowledging this, but still the global estimates of greenhouse gases emissions from rivers and streams at the global scale ignore the intermittent rivers and especially the dry phase, and only take into account the perennial rivers when flowing. Next, please. So in this project, in this work package, we wanted to analyze the effects of flow intermittency on river ecosystem functioning. And in this presentation, we will focus in the determination of the patterns and drivers of the greenhouse gases emissions in drying river networks, quantifying the contribution of the dry river beds, and also of analyzing the effects of temporal and spatial drying patterns on leaf litter decomposition. And the novelty of this study is like the wide spatiotemporal scale we are covering, because the data has been collected in different hydrological seasons to cover this variability of and this dynamism of intermittent rivers. We have been doing the sampling in different river networks, in different perennial and intermittent reaches, and we have taken samples both in flowing waters and in dry riverbeds. Next, please. So I will first explain the general study area. The study has been conducted in Europe in 6 different DRNs with both perennial and intermittent reaches in all of them. So like this, we are covering a wide latitudinal range and different climates. And on each DRNA, we have 20 sampling sites that we have been sampling 3 times during 2021. One in spring, like more basal flow conditions, then in summer during the dry period, and then in autumn, winter, after the rewetting, after the drying. And on each campaign, we have been sampling in the habitats that were present on each site. It could be like flowing conditions, dry river beds, and sometimes dry river beds with isolated pools. And in the next slide, you can see the different configurations of the DRNs. As you can see, they are all different and all having like different intermittent and perennial sites distributed all along the river network to cover like all the variability and represent the natural conditions. Next, please. Now I will present the main methodology and the first results of the part regarding the greenhouse gases emissions. Next. So to sample the greenhouse gases emissions, we have been using infrared gas analyzers connected to chambers that are the 2 kind of chambers you can see in the picture in a closed loop system. And we have been taking samples, taking measures, of 5 minutes, 5 different replicates per site and habitat. That means in perennial rivers, we could have a fully flowing riverbed in which we took the sample with the first chamber you can see in the picture, kind of a boat shape. Then during the contraction phase, we have the flowing surface is less, and we were taking the samples both in the flowing part and in the exposed riverbed on the sides. And for the intermittent rivers, we can go to the situation in which there is no flow, they are totally dry. And in that case, we were taking the sample in the dry riverbed. And for this, we use the second chamber you can see in the picture. Next, please. So we took these measures in every single site and every campaign. And together with them, we also measure a bunch of other environmental variables like air and sediment temperature, water pH, conductivity, dissolved oxygen, water temperature, and water chemistry. We also measure the discharge. and we took sediment samples either from the flowing riverbed or from the dry riverbed, from which with them we analyzed the moisture, organic matter content, pH and conductivity, and CN content. Next, please. So with all this data, apart from quantifying the magnitude of these emissions, we wanted to determine the drivers of those emissions, especially focusing in testing if they were the same or different for perennial and intermittent rivers. So we performed this modeling, including several drivers, not only including the local drivers that are the variables I told you we measure in situ, like the water temperature, sediment characteristics, and so on, but also including more distal variables like the land uses and geology in the catchment, the climatic conditions, and also including several drying metrics like the drying severity, the flow prevalence of the sites, the length of the dry periods, and also some other variables measuring the spatial fragmentation of the river network during the dry periods. Next, please. So with all these variables, we perform 4 random forest models, 2 for the flowing water conditions and 2 for the dry river beds. We perform one model for perennial rivers and 1 for intermittent rivers for each condition to check if the drivers of the emissions were the same or different for the 2 conditions. Next, please. So first, what we will show is the magnitude of the emissions we measure in the field. So here you can see the CO2 emissions of the 3 campaigns. The first one in spring, second one in summer, third one in autumn winter, from the perennial and intermittent reaches, and the emission taken on each of the 3 habitats. As you can see, the magnitude of the emissions is in the same range for perennial and intermittent rivers, both for flowing and dry conditions. I have to mention that it was due to methodological constraints, it was very difficult to measure the emissions in the pools because it's difficult to capture, to be in the field when the pools are there, because normally then they totally dry and disappear. So the replicas that we had from the pools didn't allow us to determine the drivers of emissions from the pools. Next one, please. In this one, you can see the same kind of graph, but for the CH4 emissions, where again, we can see that the emissions of perennials and intermittent rivers were in the same range of magnitude, both for flowing conditions and for dry conditions. But we have to take these results carefully because, again, due to methodological constraints with the gas analyzer equipments we were using, we were not able to take CH4 emissions from 2 of the DRNs, so the amount of data we had here is less. Next, please. And in the 2 next slides, what I will show you is the results of the random forest models testing for the drivers of the emissions. In the left, you have the CO2 emissions, and in the right, the CH4. In the upper panels, the main drivers, the 3 main drivers for the perennial reaches, and in the lower one for the intermittents. Here, what we can see is that both for CH4, for CO2 and for CH4 emissions, the drying severity was in between the top 3 drivers in the intermittent rivers, even when flowing. I forgot to say that in this slide, you have the emissions in the flowing conditions. We can see that for CO2, water pH was one of the main drivers, so in a negative relationship with the emissions. But in intermittent rivers, drying severity was also very important. Drying severity was calculated as the mean duration of the dry period. So we can see that longer drying periods increased CO2 emissions. While for CH4, we can see that water pH was also in between the main drivers, but again, for intermittent rivers, drying severity was very important, but in this case, the trend was the opposite. So higher driver severity decreased CH4 emissions. Next one, please. And in this slide, you can see the same kind of plots, But for the emissions from dry riverbeds, in this case, we have more or less the same drivers for intermittent and perennial ridges, and we can see that the climatic variables were very important for both CH4 and CO2 emissions. And interestingly, we can see that almost all the predictors show either an asymptotic or a bell-shaped relationship. For example, we can see that evapotranspiration increased the CO2 emissions, but up to one point in which the relationship turned to asymptotic. So in general, we can see that the emissions in the dry river beds are driven by a trade-off in between increasing the microbial activity, for example, with increasing sediment temperature, precipitation, or evapotranspiration, but only up to one point in which either we have a lot of moisture that can limit gas transfer, or either, for example, increasing sediment temperature, we will have a moment of water stress in which we are also limiting microbial activity. Next, please. And to conclude with the part about the greenhouse gases emissions, we did an upscaling effort trying to upscale we upscaled the CO2 emissions to the whole river network from the 20 sampling sites to all the river segments and for the whole sampling year. And to do so, we first calculated for every single ridge the daily flow and dry surface. As I said before, in the perennial rivers, we can have the fully flowing riverbed, or during the contraction and extension phases, we can have a part flowing and a part dry. And then for the intermittent rivers, we can go to the totally dry riverbed. We calculated these surfaces in a daily scale using the daily flowing, the daily discharge data coming from working package 1 and hydrogeomorphological equations. And then we rerun the random forest models I showed before, but including only the predictor variables that were available for every single segment of the river network. And with this, we calculated the daily CO2 flux rates for flowing waters and dry river beds, perennial and intermittent ridges. And with these two data sets, we were able to determine the daily CO2 emissions, taking into account the emissions from the dry and the flowing surface. Next, please. So in this slide, I will be showing only the results for two of the DRNs, the more extremes in the latitudinal gradient. In the left, you have the results from the Finnish DRN that it's in the boreal ecoregion, and the Spanish DRN in the right in the Mediterranean. And what you can see is the emissions, the summarized emissions for all the river network along the sampling year, so 2021 from January 2021 to January 2022. And what you can see in the orange area are the emissions from the dry river beds, and the blue area are the emissions from the flowing part, from the flowing surface. And as you can see, the contribution of the dry river beds in Spain, it's more than 50% of the total. But even in Finland, that it's the more perennial river network, in summer, we have a peak of the contribution of the dry river bed, and taking into account all the year, it goes up to 14%. In the right, you can see also in the small table the contribution of the dry river bed for the other river networks. And especially in Hungary, in Bukosti Beach, we can see that it's up to 77%. And taking into account the future conditions and the climate change scenarios, we expect this contribution to be even higher. So in fact, this highlights the necessity of including also dry riverbeds and non-perennial ridges in the global estimates of CO2 emissions from rivers and streams. And I think it's the last one regarding greenhouse gases. Yeah.
So then I will take over. Can you hear me well? All right. So as Nayara was introducing in the very beginning, I jumped to a different ecosystem function. We are going now to see the main effects of drying at the river network scale leaf litter decomposition. So Nayara did a very good introduction about how like the flow intermittency and drying can affect a function like greenhouse gases emissions that are really tied to bio. So it is really a function that is really, or there is a strong link between the drying effect and biochemical processes, right? So this is really clear as Nayara was showing at the temporal scale. No, when you have a river that is getting dry, then this is going to affect all those processes related to either microbial activity or the reaeration of the gas from the riverbed to the atmosphere, right? So for the decomposition is for leaf litter decomposition, flow intermittence is going to affect more or less in a similar way regarding the temporal scale, right? Because we have these scenarios of rivers that get dry and then re-wetted again. And this obviously is going to control, for instance, microbial activity and the microbial decomposition of leaf litter, no? Or even the activity of detritivores, no? If the river is dry, then obviously there is no detritivores. But this is a more still a very focus or approach that is very focused on the temporal effects of drying on decomposition. In this case, also we could mainly talk about maybe at a rich scale, at a local scale, but then we have or we can jump to the spatial effects of flow intermittence. Next slide, please. So when we talk about the spatial influence of flow intermittence, we can really see a jump from the rich scale to the network scale. No, in the case of leaf litter decomposition, this is really clear because depending on where the flow intermittence is concentrated in the network, for instance, in the upper scenario where in a river network where most of the intermittent or non-perennial rivers happen at the upstream part of the network, or the other way around. If the main occurrence of flow intermittence happens in the downstream part of the network, these locations of where drying happens is going to have a strong influence on the spatial organic matter dynamics at the network scale. So this is obviously important for decomposition because for decomposition to happen, there is a need for the resource that is the leaf litter or the organic, or particularly the organic matter in general. But this influence of spatial drying patterns is also important for the consumers, for either microbial decomposers or the detritivores. Next slide, please. So when we talk about river networks in general, the dendritic and hierarchical structure of the river networks are going to have a strong control on the spatial distribution of both organic matter or leaf litter, but also on the consumers, right? Because this hierarchical structure of the network and also the unidirectional flow of water is going to have a control on the spatial distribution of both resources and consumers. Then we have the add-on in our case of drying river networks, that within this river network we have rivers that get dry. So we have the flow intermittence, and in this case we have flow intermittence as a temporal barrier for the distribution of those consumers. No, so if we in this sort of hypothetical case in the slide for an intermittent river network, we can have a double influence of flow intermittence on the spatial distribution of consumers. On one side, those consumers or those taxa that can still be present in a river that gets dry, in a tributary that gets dry, they can be there either because they can resist the dry period or because they can recolonize the river once that the water is back. But this is not the only influence of flow intermittence Also, there is also the influence of fragmentation of the network and the isolation of certain parts of the network that are flowing the whole time, but they are surrounded by intermittent rivers. No, so taxa that are non-adapted to this flow intermittence can be isolated in the network in those regions. So yeah, as I was saying, this spatial distribution of intermittent rivers or non-perennial rivers are going to exert a strong control on the community composition, but also diversity on the consumers at the network scale. Next slide, please. So yeah, as Nayara was introducing in the beginning, and also considering this this context about also the influence of spatial drying patterns on potential consumers of live litter, the objectives of the main objectives that they want to talk about today are how live litter decomposition can change or vary at both temporal and spatial scales in the context of drying river networks, but also to assess how flow intermittence can affect decomposition through changes in the consumer community. Next slide, please. So this is just a very quick overview of the methods. So basically, as Nayara was introducing in the beginning, we also sampled over 6 European river networks in every 20 sites that we had, either perennial or non-perennial. In every one of them, we installed 3 coarse mesh bags containing popular live litter, and we replicated these decomposition experiments during 3 campaigns, no? As Nayara was saying. Like in the spring, before most of the rivers were getting dry, during the drying peak season in summer, and then in fall after the most part of the network was again rewetted. In addition to these 6 river networks that we have in Europe, we also replicated these decomposition experiments at the network scale in thanks to our select partners in Bolivia, Brazil, and Ecuador. But today I will only focus on the results of the European study cases. Okay, so this is about the logistics of the experiments. And then about how to analyze the data, we approach our function in a very similar way that Nayara was explaining. So basically, we have either factors that can be at a larger scale, like a more distal or regional scale, as can be the river network structure or the hydrological connectivity of the sites. And then we have other factors that are maybe more sort of with a local effect, no? That as can be the environment. In this case, considering also flow intermittence here, that flow intermittence, I'm going to talk not only about the temporal influence of flow intermittence, for instance, the number of dry days experienced by a site, but also the spatial drying patterns, so how much, like the percentage of the upstream network of a site that was still flowing or dry. So these maybe they are in a intermediate phase between local and regional factors controlling decomposition. And then we have also, or we are going to see the influence of consumer community, both community composition and diversity of the main taxa involved in decomposition, invertebrate communities, bacteria, and fungi. That were samples that were that our colleagues from the working package 2 were collecting during this period that we were sampling to. Next slide, please. So this is we start with having a look at the very global patterns of temporal and spatial variability of live litter decomposition, considering the 6 drying river networks together. So in this box plot here, you can see on the most left side the comparison of decomposition rates corrected or transformed by temperature of the site. The difference between non-perennial and perennial rivers. And we can see that in general, non-perennial rivers had a lower or experienced a lower decomposition rate than perennial ones. However, these differences between decomposition rates depending on flow regime were more or less modulated depending on the hydrological phase. So, for instance, the differences between perennial and non-perennial rivers were maximized during drying, obviously, because during the drying, most of these non-perennial rivers were getting dry, and therefore decomposition rates decrease a lot. No, but we can see that even after the water is back, for instance, after the flow resumption, there is still a lower decomposition in intermittent rivers than in perennial ones. So we have again this sort of legacy effect that Nayara was introducing. No, there is still a lower function rate, let's say, in non-perennial than in perennial rivers because of this previous the previous drying phase. No, so if we focus now on the box plot in the right side, here we see the temporal coefficient of variation of the decomposition rate. Okay, so during these 3 sampling campaigns that we have, how much decomposition changes over time for every site, right? So a higher coefficient of variation means that there is a higher temporal variation of decomposition at that site. So here we can see that, as we would expect, the temporal variation of decomposition was much higher in non-perennial rivers than in perennial ones. Okay, next slide. And another one. Thank you. So this is about the temporal variability or the influence of flow intermittence on the temporal variability on decomposition. Now we can see, or we can talk about the spatial variability and how this relates to flow intermittence. So here, as an example, you can see a map of the river network of the coast debit in Hungary, and you can see represented as a color gradient how high or low was decomposition for the 3 campaigns. So if you see how different the colors get for the decomposition among the 3 phases, and also you can have an idea about the variability based on the coefficient of variation that is written there. The drying phase was the moment with the highest heterogeneity of decomposition at the network scale. Okay, so if we estimate the spatial variability based on the coefficient of variation across the whole river network, the highest variability happens during the drying phase. Okay, and this is obvious too, no? Because during this drying phase, as Nayara was saying, we have this sort of a mosaic of terrestrial and aquatic habitats, and therefore decomposition rates are going to be very different depending on those either dry or wet conditions that mainly happens or coincides across the network during the drying phase. Okay, next slide. So this is not only a case for this very particular network, but we can see in this box plot summarizing the spatial variability of the decomposition rates across all river networks, that the highest variability always happen or tends to happen during this drying phase. Okay. Next slide. So here what we are going to see is which are the main drivers, or which can be the main drivers controlling this temporal variability of decomposition. Okay, so we have sort of a double hypothesis. Okay, so this temporal variability of decomposition can be either because of a change in environment, from flowing waters to dry to flowing waters again, or can be also because a change in the community. Okay, a temporal change in the community, a temporal turnover of the community, of the consumer community. Okay, so in these graphs, what you can see a summary of these results. Okay, so starting in the top left, we can see that the variability, the temporal variability of decomposition increases with the number of dry events. Okay, so we saw that with increasing frequency and also the length of the drying period, we increase the variability. Okay, following this idea of changing environment increases the temporal variability of decomposition, as we could expect. However, when we try to estimate when we try to estimate a sort of temporal variability of the sites and translate and see if this explains also decomposition, we see that there is not much relationship between our estimation of environmental variability or temporal variability with decomposition rates. So this is not very clear. What we can see in the two plots below is the connection between this temporal variability of decomposition and the changes and the temporal changes in the consumer community in this particular case for the invertebrate consumers. Okay, so in first place we can see that the temporal variability of decomposition increases with the functional beta diversity of the invertebrate community. Okay. So with the temporal turnover of feeding traits of the invertebrates, okay? And in addition to this, we can see that in the contrary, the variability of decomposition decreases when with high functional richness of feeding traits for the invertebrate community, okay? So next slide. So this, sorry, one less, well, you can leave it there. Thanks. So these two results about the influence of temporal changes in the community with the variability of the composition are really strong, because these are saying that under conditions that we have more stable communities with a high functional diversity, we have a less, or there is a lower variation, temporal variation of decomposition. Or in other words, a higher stability of the function when we have more stable communities with less turnover change and with higher functional diversity. Okay, next slide, please. So when we split these trends between the flow regimes and we separate perennial and non-perennial rivers, we can see that the relationships, this relationship between the variability of decomposition and the temporal changes in the community are stronger for the perennial rivers, or in other words, non-perennial rivers are not that sensitive to this control of the functional turnover of the community. So this might mean that the non-perennial rivers are more sensitive to this temporal variation on the decomposition. Next slide, please. So we have been talking about the drivers of the temporal variability of decomposition. Now what we are going to see here are the drivers of the decomposition rates. Okay, so which factors, either related to environment or to the communities, increases or decreases decomposition rates across the different river networks that we studied. Okay, and we separated this exploration of drivers considering separately non-perennial and perennial rivers in a similar way as Naya did. Okay, so and I forgot to say that this is also based on random forest models. Okay, so in the left side you have a variance importance sorting of the main controls of the main drivers of decomposition rates for both non-perennial and perennial rivers. Okay, so if we start with the non-perennial ones, we can see that we have four factors that were selected. Two of them were related to flow intermittence, the number of dry days, but also the percentage of the upstream network that was flowing. Okay, so both temporal and spatial components of flow intermittence. And then we have another two factors that were related to the diversity of the fungal communities. Okay, in the case of the perennial systems, we have again one factor related to flow intermittence, that is the percentage of the upstream network that was flowing. So this spatial component of the flow intermittence also being very important for as a driver of decomposition in perennial systems. And then we have other factors that were related to the local condition, the local environment, like, for instance, the nutrient concentration or the amount of carbon and so on. And finally, also a strong influence of the consumer communities, in this case, of the bacteria and the invertebrates. Okay, next slide. So to summarize and to wrap up, we can see that for the non-perennial rivers, obviously temporal, but also spatial components of flow intermittence are obviously the most important drivers controlling decomposition rates in these systems, but also the diversity and the community composition of the fungal communities. When we jump to perennial streams, we see that that there is a stronger influence of local environmental conditions, as can be nutrient conditions, but also, and this is really important, there is also, or we can see also a strong influence of spatial drying method, of spatial component of flow intermittence. In other words, of the hydrological connectivity of the network. Okay, so this is also important for perennial rivers, even if they don't get dry. It's important which part of the network is above a site is getting dry or not, and for how long. And lately we have also the strong influence of both the diversity of bacteria and invertebrate communities, and also here just realize that biodiversity is important to control both decomposition in non-perennial and perennial rivers, but we have a switch between which communities or which taxa are controlling the decomposition the most in one place or the other. In non-perennial systems, we have fungi with a more important role. In perennial ones, we have more bacteria and especially invertebrates exerting a more important role on decomposition. Okay, next slide. So these are just a little bit of some take-home messages to wrap up the presentation of today. So starting or going back to greenhouse emissions, as that Nayara explained so well. So basically, we need to consider that intermittent ridges can emit as much, at least as much, either CO2 and methane as perennial systems can do. But however, we need to model these emissions from intermittent ridges in an independent way than we do for perennial systems. And this is especially because of this drying legacy effect that can affect CO2 and methane emissions even after the resumption of surface flow. And also, as Nayara was saying, and this is one of the most important things, these results are a very important evidence that we need to include the emissions from either intermittent ridges and dry riverbeds for global estimates. Otherwise, we will be at the risk of underestimating severely the global greenhouse gases estimation from rivers. And in the case of leaf-litter decomposition, next slide. There we go. Then some of the main take-home messages that we can see is that flow intermittence can decrease decomposition rates, but increase temporal and the spatial variability of the decomposition. We have seen that decomposition rates are mainly explained by these temporal spatial components of flow intermittence, but very importantly also by consumer biodiversity. We have seen also that the temporal turnover and functional diversity of the consumer is strongly important to control the stability of the function at least of leaf-litter decomposition, and potentially that non-perennial streams might be more sensitive to changes in biodiversity to regulate this stability. So with this, I am also over. Next slide. The last thing that I really would like to do is to thank everyone for joining our webinar today, and very especially to thank every single team from Work Package 2 and Work Package 3 that were involved in the sampling of both functions and biodiversity, that were like really helpful and make all of these results possible. So thanks everyone.
Okay, thank you, Nayara and Ruben, for this great webinar. Does anyone have a question for them? Okay, Pierre, please go ahead.
Hello, can you hear me?
Yes, we can.
Hello, hey. Okay, so if I can just speak, thanks a lot for the talk. It was super interesting. It's for Ruben. I was wondering, when you look at the effect of what you assume to be hydrological connectivity with a percentage of upstream flow, did you in some way correct for the abundance of coarse particulate organic matter? What I'm thinking is that if this is correlated to position within the network, I assume it's also correlated to land cover and the amount of accumulated organic matter. And if you have a sense, if you haven't done it, if you have a feeling as to which might affect your response variables more. Thanks.
That's a great question, Pierre. So, well, I mean, two things, no? Like the way this is estimated, this upstream flowing percentage of the network is only based on, obviously, on if reaches upstream of a site were flowing or not for a certain period, and just considering the percentage of the total upstream network of that site. So from 0 to 1, if the whole upstream network was flowing or not. But as you said, this is one thing, no? And another thing is how much organic matter can be accumulated because of this connection or disconnection of the flow. So the only thing that they can, obviously, this might have an influence. The only thing that I can tell you now is that in what the results that they have seen, or that I have shown, better to say, are based on random forest models. So in these random forest models, the accumulation of organic matter was also included. So then maybe this might influence the response that we see of this upstream connection of the network. But yeah, like if we only look at the interaction or the relationship between decomposition rates and accumulation of organic matter stocks, is not that important as a driver itself. At least not, okay. Maybe for certain networks is actually quite important. In general, maybe not that much. And this obviously is, it's easy to explain in a way because we are putting our decomposition rates are based on little bags that we bring to the river, independently of which organic matter is there or how much, no? So then by doing this sort of artificial experiment, we are forcing decomposition to happen independently if there is any organic matter or tons of it, no? But yeah, you're right. This is one of the main things that we want to explore further.
Okay, cool, thanks.
I also have a question for both of you. Do you expect biodiversity to affect stability of other ecosystem functions beyond decomposition?
Nayara, do you want to jump?
Yeah, I mean, regarding the greenhouse gases emissions, we have performed also another experiment in which we have incubated sediment samples that we took either from flowing water or from dry river beds, like in the same spots in which we measured directly. And we have measured the CO2 and the CH4 emissions from those samples, from which we have also determined the microbial communities by DNA metabarcoding. So we expect the drying pattern also to affect microbial communities and be linked with the emissions, but we are still working in this data. But yeah, I mean, I totally expect a relationship.
Thank you.
Yeah, we have another function that we didn't talk about today, but it's about the stream metabolism. So I think that we have some colleagues from Cantabria also here. So if they want to jump to the discussion here about how biodiversity influences stream metabolism, I am not an expert on this, but yeah, I think this relationship between the ecosystem function stability and changes in the community. Yeah, I think I will expect to change considerably from function to function, because there are some functions that are more strongly linked to local communities, as can be like decomposition, because obviously decomposition happens in local places, no? and stream metabolism, for instance, is something that happens at a much higher or bigger spatial scale, and maybe then, biodiversity changes are less, or is less sensitive to changes in biodiversity. But maybe colleagues from Cantabria can jump in here too.
Yes, Maya, please.
Yeah, so we didn't find any direct relationship between the biodiversity and river ecosystem metabolism. We did find more not an indirect effect of biodiversity in gross primary production. So the biodiversity of algae and diatoms and macrophytes, they affect the quantity of chlorophyll A, and then chlorophyll A and light in each day are what affect gross primary production in a river segment. So that would be the relationship. There was no relationship between heterotrophic organisms and respiration, but almost all of the respiration came from autotrophic organisms also. So there might be a complex relationship there.
Teresa?
Yeah, hi. Can you hear me okay?
Yeah.
Yeah, really nice presentation from you both. Thank you. I just have a question for Nayara. You did the upscaling for CO2, but I was curious if you did it for methane, or if not, maybe what your expectations would be for the methane emissions.
Yeah, it's interesting question, because the thing is that for methane emissions, as I said when I presented the general plots with the magnitude, due to methodological constraints with the greenhouse gas analyzers, we only have data from 4 out of the 6 DRNs. So in fact, although the random forest models were performing pretty well, they were not robust enough to be able to use them to upscale and to predict for the other segments. So in fact, it would be super interesting to do like the same approach of the upscaling of the CO2 for the CH4. But in fact, for this, we will need to have more data to be able to do it. And also, I didn't have a lot of time to put a lot of attention on that, but for CH4 emission, there were also local drivers like the sediment temperature that were very important. And those we don't have this fine local scale data for all the river segments. So to summarize, we need more research on CH4 to be able to do the upscaling.
Okay, thank you. Is there any other question? Pierre?
Hello again. I have one last question. It's again for Ruben. I think maybe I missed it, but I was wondering if you looked in detail at the effect of the timing of drying within the year between seasons. And I'm asking because I'm looking at leaf litter decomposition in alpine streams here, and the drying patterns are kind of opposite to what you'd find, I guess, in Mediterranean systems where it dries a lot in winter. And it seems that we have some effects of timing of drying on shredder communities, which probably cascade down to organic matter decomposition. I was wondering if you looked into this, and if you did, what you found.
That's a good point. So yeah, I think so. We have, well, maybe this is we can explore this further when we have also the data, or when we explore also the data from select countries from South America, because obviously then the times are going to be very different for as well. In our case, most of the networks, except the one in Finland, have, I think, a very seasonal pattern of drying, mainly happening during summer. So we see mainly this sort of negative effect of drying on decomposition. In Finland, that is maybe the most different network in the Finnish networks that are the most different to the rest of the data set, is, as you said, the place where we see the less influence, or the less influence of drying on decomposition. I think this is also related to, obviously, to a lower degree or a lower intensity of drying happening there. Not sure if, I think also in Finland, the drying also happens mainly in summer, I would say, but for sure decomposition is less sensitive to flow intermittence in this network.
Okay. Okay. Cool. Thank you.
Is there any other question? Okay. So if there are no more questions, I would like to thank you all for attending the meeting, and thank you again, Ruben and Nayara, for your presentations. Next webinar will be on January 17 from 11 to 12. If you would like to attend, we will soon send you the link and publish it on the social media. So, thank you. Have a nice day. And
Thank you, everyone.
Thank you.
Thank you.
Thank you. Bye. Thank you.
Bye.
Thank you. Bye-bye.